Morning. In previous class, we studied about development of anther. Okay, there we have already studied different layer of anther, tapetum layer. Today we study about the development of pollen grain. That is microsporogenesis. Okay, today we study about the development of microspore. That means pollen grain. In which way the pollen grain is developed inside the anther. Okay, in the structures of anther, there we know the sporogenous tissue. From the sporogenous tissue, they are from develop the microspore mother cells, which is also known as pollen mother cells, and their short form we can write as MMC or pollen mother cells. That means PMC. Okay, microspore mother cells and pollen mother cells. Understood? The sporogenous cells may function directly as the microspore mother cells or undergo a series of meiotic divisions. Okay, we are all know that the gametic cells are haploid. Why? Because the gametic cells are developed through meiotic divisions. During the meiotic divisions, the developed cells are all haploid. That means n number. Okay, the pollen mother cells are polygonal in shape and are closely packed. Okay, as the anther increases in size. When anther increases in size, the pollen mother cells are continuously matured and converted into pollen grain. Okay, the development of pollen grain, the process formations of microspore pollen grain, that is known as microsporogenesis. So position of microsporogenesis inside the anther, where from started? Now from the sporogenous tissue. Okay, that sporogenous tissue is developed from the developed inside the anther. Okay, now we start the development of microsporogenesis. Okay, bilkul, that microsporogenesis occurred through the meiotic divisions. The microspores are formed by meiotic divisions of MMC, that is microspore mother cells pollen mother cells. Okay, each undergoes meiotic divisions and forms 4 haploid pollen cells or microspores. Okay, after then they are going to develop the pollen tetrad. Okay, what do you mean by pollen tetrad? Initially the microspore exit together in a tetrad conditions called microspore tetrad. Okay, or pollen tetrad, enclosed within a common wall of cellulose. Which wall? No, it is component of cellulose. The pollen mother cells, that means when it convert matured in pollen grain tetrad, then there presents one wall. That wall composed of callus. Okay, callus, not cellulose, callus से बना हुआ होता है. ठीक है. Gradually the microspore separate from each other, develop an outer covering around each of them and then mature to 4 pollen grains. That is tetrad pollen grains. They are going to develop 4 mature pollen grains. In this way, that pollen grains formations are completed. Now we are going to understood through a diagram. Okay, suppose inside that MMC microspore mother cells, that is the microspore mother cells. Okay, when start the first meiotic divisions, that microspore mother cells divide into 2 shades. Okay, divide into 2 cells. That is the meiosis 1 divisions. Okay, meiosis 1. We are all known that the meiosis cell division have 2 phases, meiosis 1 and meiosis 2. Okay, that first meiotic division, meiosis division, that microspore mother cells which is present in 2 n conditions. Okay, 2 n conditions. That means diploid. After meiosis divisions, it is converted to two cells. Then that second meiotic divisions, it is developed into four cells. Okay, developed into four cells. Okay, four cells. Okay, this is the known as pollen tetrad. क्या कहते हैं? Pollen tetrad. Here that cells are in presence in haploid. Okay, that is called, here it is called pollen tetrad. Pollen tetrad. Okay, pollen tetrad. Okay, that pollen tetrads gradually is developed, and here from matured, here from develop the mature pollen grains. Here from develop the mature pollen grains. That pollen grains have two layers. Outer layer is called exine and inner layer is called intine. Mature pollen grains. Understood? Okay. That is the pollen grains. That pollen mature pollen grains. They are present two type of nuclei. One is generative nuclei and another is pollen tube nuclei. Okay, and they are present some pore like structure that is called the germ pore. Okay, suppose this is a mature pollen grain. Okay, this is called exine, outer layer exine. Okay, and inner layer is called intine. Okay, they are present two nuclei. Okay, one is generative nuclei, generative nuclei. Okay, okay, yeah, nucleus initially generative nucleus. Okay, and another is pollen tube nucleus. Pollen tube nucleus. Okay, one is generative, another is pollen tube nucleus. When pollen grain enter into the stigma, after then that germinations, there occur the germinations, that pollen tube nuclei helps to develop the pollen tube. Okay, and generative cells therefore develop the antherozoids. Okay, this is the structure of pollen grains. Okay, actually that pollen grains developments, when developed, they have two types. Okay, their development is two types. One is successive type divisions. Developments, here the type of divisions, the type of divisions is two types. One is successive type division and another is simultaneous device. ठीक है, simultaneous type of divisions. Okay, according to their division systems, this is the general process. Okay, this is the general process of microsporogenesis. Okay, क्या होते हैं microsporogenesis में ना, microspore mother cells divides होते हैं, first meiotic divisions. After the second meiotic divisions, they have developed the pollen tetrad. That pollen tetrads, when matured, is converted to pollen grains. Okay, but the development systems is two types. Number one is successive. Successive type of divisions, successive type, and another is simultaneous type. Okay, simultaneous type. Okay, simultaneous type. Okay, in case of successive type cell divisions, there have some steps, but process of division is different than the simultaneous types. Okay, now I'm going to start the successive type of divisions. ठीक है, successive type divisions. Here same first is microspore mother cells, which is present in 2n conditions. Okay, after then that developed into two cells, divide cell division takes place here, and two cells are developed. Okay, that means successively divided into two cells. Okay, next that two cells are continuously divided. Okay, that is the meiosis 1, meiosis 1, and here start the meiosis 2. Okay, here after then develop the fourth nucleus. That is the haploid. Okay, haploid, it presents in haploid stage. Okay, this is the successive type of cell divisions. Okay, that successive type of cell divisions occur in generally monocotyledonous plants. Most important point, where occur the successive type of cell divisions? In case of example is monocotyledons. Monocotyledonous plants. In case of monocotyledonous plants, that successive type of cell microsporogenesis occurred. Successive type of divisions. That microsporogenesis occurred through successive type of divisions. Okay, and next one is simultaneous type of cell divisions. Okay, here same that microspore mother cells, that is the microspore mother cells, is present in 2n conditions. Microspore mother cells here also divides the cell. But what is the difference between successive and simultaneous? Here the after meiosis 1 stage, here divide two different type of cells. But here the cells are present in a 4. Okay. This is the difference. Okay, after then here from develop, that is meiosis 1, that is meiosis 1. After meiosis 1, that means meiosis in meiosis 2 stage, they are developed in this type of division. Okay, okay, next that is develop the tetrad. Here that cells are present in that way. They are here we observe 3 cells and another cell is present behind that 3 cells. Okay, that is the simultaneous types of cell division. That simultaneous type cell divisions occur generally dicotyledonous plants. Dicotyledonous plants. Okay, this is the simultaneous types of cell divisions. Okay, but that microspore tetrad, when it is developed in the development process in presence in different plants according to their species. Okay, different plant species have different type of development process. Okay, next the success in case of successive. Why it is called successive type of cell divisions? Successive types because there are difference. What is the difference between successive and simultaneous types? In case of successive, they are now occurred. Each nuclear division is followed by cell wall formations. In case of successive, cell wall formation, here cell wall formations occurred. But in case of simultaneous type of cell divisions, their cell wall formations is not occurred. Okay, and that first stage is present in dyad condition, after then tetrad. Okay, the first nuclear division is followed by cell wall formation, resulting in the formation of two haploid cells. That is the two haploid cells. Okay, after meiosis 1 divisions, two haploid cells, which is known as dyad. That is called dyad. D-Y-A-D. Okay, understood? That is called dyad. Okay, that stage presents in successive type of divisions. Okay, and after then it is also followed by wall formation, and it results in a tetrad, that is the haploids, that haploids. Okay, tetrad conditions. Okay, tetrad conditions में प्रेजेंट रहते हैं कौन सा ना, ये डायड के बाद आता है कौन सा स्टेज ना, टेट्राड, tetrad. Okay, why? As present four nuclei haploid cells, tetrad. Successive wall formation is commonly found in monocot type of cell division, monocot type of plants. Okay, that is the successive type of microsporogenesis according to their division process. And next is simultaneous types. Again I repeat that process. क्या होते हैं ना, इन दिस केस meiosis 1 is not followed by cell wall formation. Here cell wall formation is not occur, but nuclei is divided, their presents to nucleus, but wall formation is not occurred. That is the difference between successive type and simultaneous type. Understood? So instead of microspore dyad, a binucleate cell with two haploid nuclei is formed. Here dyad stage is not present. Instead of dyad stage, here binucleate cells form. Okay, after then the cell wall formation occurs simultaneously. Cell wall formations occur in between the four nuclei, resulting four haploid cells, that is the microspore. Okay, when occur the cell wall formations? After the two stage cell nuclei, that is called dyad, but here it is called two stage cells, two stage nuclear cells, because here have no any cell wall formations. Okay, after the two stage nuclei forming stage, after then develop the four nuclei shell, four nuclei presents here. Okay, the wall formations is centripetal. Here wall formation is centripetal, not successively. Here wall formation is centripetal. Okay, starts from the lateral wall and meets in the center of the cell. Okay, lateral wall start the lateral walls and meets in the center of the cell. Okay, these kinds of cytokinesis, meaning of cytokinesis, जो वॉल फॉर्मेशन हो रहे हैं, ठीक है, सेल की डिवीजन जो हो रहा है यहाँ पे स्टार्ट, इसको क्या कहते हैं, साइटोकाइनेसिस. कैरियोकाइनेसिस मतलब डिवीजन ऑफ न्यूक्लियस. ओके, यू आर ऑल नोन इन ऑलरेडी स्टडीड इन क्लास 11. ओके, दिस काइंड ऑफ साइटोकाइनेसिस may lead to the arrangement of microspore tetrad in various ways. Simultaneous wall formation is commonly followed in dicotyledonous plants. Okay, in dicot plants, simultaneous types of microspore formations presents. Okay, that is the difference between simultaneous types and successive types of microsporogenesis. Understood? Okay, the arrangement of microspore tetrad कैसे अरेंज रहते हैं, एंड माइक्रोस्पोर टेट्राड के अरेंजमेंट क्या कैसे रहते हैं. द माइक्रोस्पोर ऑफ ए टेट्राड आर सेपरेटेड फ्रॉम ईच अदर बाय कैलोज वॉल. एकदम स्टार्टिंग में हम ये चीज पहले ही बता दिए हैं, जो वॉल जो फॉर्मेशन होते हैं कैलोज वॉल फॉर्मेशन होता है. ठीक है, पर पार्शियली sorry, that is the formations of callus wall formations होते हैं. callus wall जैसे formations हो जाते हैं, क्या होते हैं, there is no connections between the microspores of the different tetrad in same anther loculi या anther माइक्रोस्पोर सैक के अंदर क्या होते हैं, ये जो टेट्राड जो रहते हैं सब एक दूसरे के अंदर क्या होते हैं, जो जैसे वॉल फॉर्मेशन हो जाते हैं, क्या हो जाते हैं ना, सब अलग अलग तरीका से वो माइक्रोस्पोर ये फॉर्मेशंस हो जाता है, दैट इज पोलेंड्रियन फॉर्मेशंस हो जाता है. ठीक है, टू लेयर्स, दैट इज टू लेयर्स पोलेंड्रियन फॉर्मेशंस हो जाते हैं. ओके, द अरेंजमेंट ऑफ स्पोर्स इन ए टेट्राड्स may show variations. It may be tetrahedral, isobilateral, okay, linear forms, okay, that divisions also occur by T-shaped divisions, etc. Okay, of this tetrahedral and isobilateral arrangement are the common type of डिविजन्स ठीक है क्या होते हैं कॉमन टाइप्स कौन सा कॉमन टाइप कौन सा है इट इज मोस्ट इंपोर्टेंट फॉर नीट एग्जाम tetrahedral and isobilateral is the common type tetrahedral and isobilateral is the common type okay and some another types presents which is known as aristologia elegans all the five types of tetrad arrangements are present yeah it is also that note that point aristologia okay which plants in case of aristologia a r I s aristologia l o c h I a aristologia aristologia जो प्लांट्स जो होते हैं ठीक है उसमें क्या होते हैं ना all five type of tetrad arrangements are present okay that clear in next class we study about the megasporogenesis okay megasporogenesis okay okay thank you